All right, today, webinar number 2 of our series, of our diagnostic series, is advanced tools for plant disease identification. Advanced tools meaning we are looking at what happens once we get back to the office. Webinar 1, we looked at field tools, what we're going to do in regards to bringing out into the field. We looked at a bunch of different tools. Some of those are going to carry back to the office, some will not. We're making 2 assumptions. Either you have been on that field visit, and if you've been on that field visit, you've collected a lot of information, probably more information than you would need, just in case. Option 2 is a walk-in. A walk-in can happen 2 ways. A walk-in can be left at the desk with no information, or the walk-in can actually speak to you and give you some verbal information. So we're going to kind of toggle back between those. And this is where I'm going to need you to chime in if I'm not clear. So I'm going to do some generalizing and I'm going to kind of go back and forth. But the assumption at this point, our advanced tools, is that we're back at the office and we have a sample sitting on our desk. Okay. Questions? Okay. So let's assume you went on the actual field visit. When you're on a field visit, you've got a stakeholder over your shoulder. You've got maybe a commercial grower who needs answers now and they're panicked, or you may indeed have a, just a homeowner who has a lot of questions and maybe is talking about a lot of different things. If you know that person, you may be getting chit-chat in between. So now you're back at your office and all is quiet. And this is the point where you review the information you've collected. If you've taken photos, you've looked back at those photos, you have a chance to think a little bit more clearly than you did before. So this is where you're going to review all of your information. If it's a walk-in, you have to question now, what got dropped off? What sample is actually physically there? Do you have the right sample? That's a question. You also you also have the possibility that the wrong sample came in, that it was just dropped on a desk. Maybe you were out and the sample just arrived, or maybe that person came into your office and you were able to talk to them. So if you were able to indeed talk to that, that stakeholder, you have to ask all the same questions that you asked when you were on the field visit and see if you can make heads or tails of what the problem is. And you know, as well as I do, that our stakeholders have probably different terminology than we would use if we were talking to each other. So to be able to ask repeated questions and to drill down and get to the root of the problem. And that's where our field visit checklist, that PPFS Gen 15 that we reference, I'll put it in today's references as well. That's where that's going to come in, is to help that person or to help you remember to ask all the questions that you may need. Another option is, or the plant diagnostic lab forms. If the sample just appeared, you've got to make a lot of decisions on whether it's something like a leaf spot that's obvious. Did they bring a proper sample? Most of the time they don't, unfortunately, and you might have to make a phone call. So just kind of going through those. Now, we are always going to begin with the assumption that a sample might go to the plant disease diagnostic lab. So you have to have a sufficient amount of information, and you have to get that information recorded. So I am going to share my screen again and go back to the diagnostic lab or to our website. Okay. It's going to take just a second. Usually takes a second for you to see the screen that I'm sharing. So Kim, can you help me out? Yeah, there we go. Sharing my screen. So here we are back at the plant pathology homepage, plantpathology.ca.uky.edu. And you can just go into extension right here. If you're seeing my cursor, click extension. And then there's this entire menu and plant disease diagnostic lab here. So if you go to the diagnostic lab page, right here on the bottom, County Extension personnel, plant disease identification forms available here. This is requiring a login now. This is something that we've done that's new. So you would click login right here, and you just are going to log in with your UK credentials. So it's nothing new, and I have already done that. This is where it takes you. All right, these are your diagnostic lab forms. You can use either of these forms, and this is where you're going to collect information. We need all of this information every single time, no matter what. This is what you're going to need. So ask these questions in particular. So it's a single page. If it's an ornamental, we're going to need more information, and there is a second page. This is the one we used to tell you to print on yellow paper. It actually used to come on yellow paper, if any of you remember back in those days. Then at some point we told you to print it on yellow paper. Now we would just be very happy if you would use it at all. This gives us basically information about the plant and about the site. If you're on the site visit, you're going to collect all of these things. If it's a walk-in or if something gets left on your desk, you're going to have to go back and get this information. And through the next few weeks, we are going to see why all of these things matter so much. So has everybody seen this page? Has anybody gone here yet, or do you, how do you do your diagnostic forms? We used to mail them out, if you recall. So we don't mail them out anymore. They're just online. And again, okay, good. Thank you, Camille. So anyway, you're going to make the assumption that there may be a sample going to the diagnostic lab. If there is going to be a sample to the diagnostic lab, you need all the information and you need proper samples. Remember last time we talked about packaging. So you need all of those things. You may end up not having to send a sample to the diagnostic lab. That's okay. You've at least dotted your I's and crossed your T's. So that's going to be there for you. The requirements to the diagnostic lab will also help you. So you can start training your growers and your stakeholders to give you what you need to make that diagnosis of any kind. So questions there? Okay, so moving on. In addition to the diagnostic forms and the information, rather, on those diagnostic forms, images always help. You don't necessarily send those images to the diagnostic lab unless they ask for them, but it's good to at least initially have them. One thing that we put in the resource guide last time, remember the Take 5? Taking photos and how to take photos, what kind of photos to take for us. I'll reference that again. So collecting images, remember there are 5 requirements for the images. We want the entire site, give us an idea of the topography, surrounding plants, the condition of those surrounding plants, etc. So number 1 is a wide angle view. Photo number 2 is the entire plant. That gives us a little bit more resolution. What does the entire plant look like? We're looking for thinning, we're looking for dieback, we're looking for lichens and leaf color and all of those things. That usually helps us eliminate a lot of abiotic issues. So the first 2 photos. So we need that photo. Third photo is we are looking for cankers or any kind of branch or stem lesions, anything like that. So it would be a close-up or 2 of something really specific that's just not right. So you're kind of looking down, following back like a dead branch or a dying or unhealthy branch, following that back, seeing if there's anything in common there that maybe needs a second look. So I just kind of generalize and say cankers. So that's photo number 3. Photo number 4 is the soil level. Always take the soil level right there at the ground, the crown of that plant. I want to see what kind of weeds are there. A lot of times weeds will tell us if there's compaction. If there's goosegrass, for instance, we've got compaction. If there's dollarweed, we have a water issue. Maybe it's very wet. This is how we usually see if there is maybe a possibility of herbicide damage. If we see dead weeds, then maybe there was a recent herbicide application. So a lot of things we can see. We can also see string trimmer damage, mower damage, if there's a dog chained to the tree. When I was first hired, I remember there being like a little hatchet, and a kid had been whacking on the bottom of the tree. So right there, that soil line tells us a lot more than we admit. So photo number 4 is always the soil line. And then photo number 5 is going to be any leaf images that you want to send. So my rule is 5 pictures, never more than about 6 or 8 pictures. They're never going to be zipped to us, and they're going to be limited. So, you know, we talked earlier about email overload. So don't overload emails, and please don't zip it, because if we're driving, that we usually have a problem opening zip files from our phones. So a limited number of pictures, but enough pictures to tell the story. I usually use trees and ornamental trees as examples, just because they have all of the extreme symptoms and more variation than anything else. So that they're always a really good example that I give, but this applies to all crops. So in regards to communicating with a specialist, or when this sample comes in, you have 2 options. You can work on it yourself and prepare for the diagnostic lab sample to be submitted. They may or may not require pictures. We don't send photos to the diagnostic lab every time, unless we print them and put them in the box, just because they're dealing with thousands of submissions every season. So we can't overload them. You do have the option, though, of communicating directly with me or one of the other specialists. I welcome photos. These are my rules. Other specialists may have a little bit different rules, and entomologists, of course, don't require all of that information. So getting the information in a photo, it's okay to message me a couple of photos and say, okay, what next? What do I do next? So especially new agents who need a little bit of guidance at first, not quite confident, feel free to message me that way. I prefer email over text, because that way I can come back to my office where I have a really big screen and I can zoom it. So you're going to attach the photos, don't embed them. iPhones in particular like to embed, unless you check the option of attaching. And that gives me the ability to zoom as well. If you embed, it's a low resolution photo that I cannot manipulate. Questions there? I know sometimes I'll get emails and then I'll kind of reply and ask you to resend it as an attachment, and that's just so I can zoom a little bit better. Okay, so backtrack. Chime in anytime, unmute yourself, and ask questions. Otherwise, I'm going to go on, but don't hesitate to back me up. So you've either done the field visit or you've had a stakeholder walk into your office. You still need all the same information, no matter how that sample came. If it's not obvious, you definitely want images, especially if it's a walk-in. You want kind of the big picture as well as the narrowed in. And then you are going to examine those photos on your own. You may or may not send them to me, and you will either print them if they're going to the diagnostic lab, or once you communicate with the diagnostician, then that would be the option to mail them. So, so now you've got the information there. You're evaluating that information. The next 5 webinars, we're going to go through the specifics on what to look for, what kind of structures to look for. As for the sample, what samples do you actually take? Which ones do you need brought to you? Which ones do you go to the diagnostic lab? And how do you start? My first rule is that you need a range, everything from symptomatic to healthy. You've got to know what healthy looks like before you can decide what unhealthy looks like. That's a fabulous way to start. We see more and more specialty crops here in the state, going from vegetables to fruit to a lot of different ornamentals, commercial and residential. So you've got to know what healthy looks like. Otherwise, we just lose track with some of these specialty crops. So at least 3, at least an extreme symptom, a moderate symptom, and healthy. Dead is too late. Okay, dead sticks are always going to come to the diagnostic lab or to me or to your desk, and there's just no way to tell. our saprophytic fungi move in. We can't tell anything by a dead branch. There has to be some living tissue in there. In regards to cankers, you've got to be able, let me see if I can get a sample here. You've got to be able to find a margin that is going from, I don't know if you can see it. Let's see, let me spin it around. You've got to be able to go not just from the canker itself, but you've got to be able to move back into healthy tissue and find the margin of the canker underneath the bark. And that is going to tell us, so the sample, if it goes into a microscope slide, it will come in at that margin. So dead is too late. So this is fire blight actually. This dead is too late. We're not going to get anything useful out of this actual canker. So we move on to canker margins. Okay, and as we work into the next few webinars, we'll actually talk about dissecting some of these samples and to know what to look for. So we're going to kind of touch on that later. So dead is too late. Even in an extreme canker, you want something more. So we always tell you 6 to 12 inches below. And sometimes you may want a little bit more than that. This is a really big branch. If you are going to maneuver this one, you can cut it in pieces and put it in a box if that's going to come to us. Something like this is just going to get you a reply from the diagnostic lab or from me that says, go back and collect more samples. So dead is too late always. Don't forget that part. And that's always kind of hard when you've got a walk-in sample that come in with something dead and you turn that stakeholder around and they're usually not real happy with that. We understand, but there's just nothing that we can do from there. So samples for you, for communication with me, or for the diagnostic lab, you need at least 3 extreme symptoms, moderate symptoms, and healthy. That's how you're going to get an idea. Okay, dead is too late. In regards to any sample, 1 leaf, 1 branch is rarely going to tell you what we need to know. We need to know how much of that plant or how much of that planting is affected. So you need multiple samples. If you're going to send leaves, send 10 leaves. If you're going to send cankers, send about 3 or 4. If you're going to send seedling plants, send 5 or more. So multiple samples, because if we don't have a sign, a fungal sign, for instance, if we don't have the actual pathogen on the one that you sent, then we're going to say no pathogen found, and where you may really have disease out in that field, that 1 sample was not sufficient. So multiple samples, when you're collecting, get multiple samples of each symptom, and that will give us better ideas. Okay? Questions? All right, so now we're going to start talking about what you do. So you're prepared to send to the diagnostic lab if necessary. You're prepared to communicate with me if necessary, but this workshop and this whole series is about you, in fact, doing what you want to do or what you can at your office. It saves a lot of time, especially at this time of year. There can be a 3-week turnaround at the diagnostic lab. When it's a commercial grower, we need to move quickly. We at least need to eliminate the obvious. So if you are collecting samples, if it's a commercial grower, we really like it to go to the diagnostic lab at some point or right away. But in the meantime, you can be working on samples. Option 2 is that you might want to try your hand at it. If you can't find anything, then send it to the diagnostic lab. So those are kind of your 2 options, which means you need sufficient samples for the diagnostic lab and you need the ones to play with for yourself. So you need twice as many samples in that case. So don't forget, you cannot mutilate a sample and then box it up to send to the diagnostic lab. That is not going to work. So have enough samples that you've got your own. You've got yours in hand and you can work with it. Okay? So, of course, doing your own detective work gets you, again, a quick turnaround, and it gets you experience. If nothing else, you can do your detective work, take your best guess, and then wait for the diagnostic report to come in. And then that way, that's how you learn as you kind of build your skills. So let's talk about some tools. We're supposed to be talking today about advanced tools. I'll give you one that's maybe not quite so advanced, is a moist chamber. So this is one of my favorite tools. This is a deli container. I use a lot of deli containers. And just anything with a lid. Anything with a lid. Ziploc bag works as well, but I like something kind of rigid. Moist chambers will induce sporulation with a sample, and by increasing sporulation, you're able to see. When it's hot and dry during the summer, your fungal pathogens in particular are not sporulating. It's harder to see what you're looking for. So 24 hours or 12 hours in a moist chamber is going to induce that sporulation, and you'll be able to see the signs of that pathogen. So this is one thing that's always a lot of fun as well. If you've ever taken a plant disease diagnostics class, you've done this. So I'm going to use this little foil container, and I save these. So these come out, they go through my dishwasher and then make it to my office. So what I do is I come in with a paper towel. This one is just kind of left over. And you line the bottom with a paper towel, and then you put water. I'm going to make a mess here on my desk. You put water to wet the paper towel. Very important is that you squeeze that out. So what I like to do is I squeeze the water out and pour it out. So you've only dampened this paper towel or two, okay? If you've got a sensitive leaf, then you might want to put ink pens or pencils down there and suspend a leaf on top of that so it's not touching the wet paper towel. Toothpicks, whatever you want. I don't know, it's kind of glary here. I don't know if you can see. There's my wet paper towel. And then putting your sample in. Putting your sample in to induce sporulation. So here, and then putting the lid. These moist chambers, here we go again with the glare. These moist chambers can sit just on your desk at room temperature. And usually if you do it one day, you can come look at it the next day. It usually only takes about 12 hours. After 24 hours, you're going to start seeing the growth of saprophytic fungi, and that is not going to give you a diagnosis. So it can't stay too long. Once you have a sealed moist chamber this way, so this one you just fold over. It doesn't matter what kind. This is a blue cheese container, again, comes out of my kitchen. But you're going to have a 98% humidity in this case. And 98% humidity should indeed induce sporulation on anything that you need. So right on your desktop, there's nothing toxic here, so it's okay to indeed leave it on your desktop. When you come back in the next morning, you can take a look. So moist chambers, absolutely critical. I've got a whole stack of these things all over my office. They come in different sizes. This one was cookies, I think, for something that happened in the department. And they always just kind of hang around. You can wash them out and use them again. In desperate times, you can use a Ziploc bag. Just make sure that you've kind of puffed it up and left enough air in it so that you don't have a wet, soggy sample. You don't want a soggy sample. You just want that humidity. You're creating that humidity chamber. Okay? Questions there? Another tool, a really high-tech tool that we use are good old phone books. Unfortunately, we still receive phone books at my doorstep. And what do you do with a phone book? Well, we press leaves in phone books. This one, this is from a hemp planting. Let's see if I can show you this. There's a hemp planting. I have pressed leaves there, and this gives me flat leaves. I don't always need the paper towel. Those were Kim wipes. But here I've got, let's see if I can get this. So I've got samples, and this keeps it nice and flat. We're gonna zoom for you. Try to zoom. So this gives us a flat leaf. So any book works. I've used valuable books, unfortunately, and you can put paper towels if it's a valuable book. If it's a phone book or similar, then you can just go straight in. Don't forget, though, if you have multiple samples, then to put a Post-it note sticking out on which ones it is if you have multiple samples. So don't think you're going to remember, because you don't. In fact, this phone book I found on my shelf, it's strawberries, but I don't know what's going on here. And I forgot to label them, so don't do as I do. But phone books and deli containers are some of the best tools that you can have in your office, and that will get you pressed leaves. If you're going to send samples to the diagnostic lab, it is always really great to press the leaves. That way they make it there without crumbling. Once a leaf dries out, you don't want to, you can't really flatten it back out. What happens when a pressed dried leaf gets to the diagnostic lab is they rehydrate it. You can do the same. You can put a pressed leaf into a moist chamber and rehydrate it, and that will induce sporulation as well. Once you've sporulated, once a sample comes out of the moist chamber, you cannot send it to the diagnostic lab, okay? This is for you to play and for you to explore. Most of the time it gets you answers. Questions? Okay, so now let's look at actually some better high-tech tools. So remember, first we started with the big picture. We got some ideas. Let's say we have a sample that we want to explore. And one thing all agents should have is the handy dandy Dino Light. About 7 or 8 years ago, Dr. Palmer bought Dino Lights for all of the counties. Not many agents really like their Dino Light. And this is a little handheld microscope with a USB port. Okay, with your Dino Light, you received this stand. Okay, this is a very wobbly stand, as you can see. It is not our favorites, and unfortunately, we just have to take what we can get. I want to show you, though, I bought, let's see, you can see, we'll go to the Dino Light site in a minute. I bought a better stand. This is about $100. I am going to switch in a second. We're going to go to the Dino Light website. So Dino Light Pro, let's see. Dino Light Pro is the one, I don't know if you can see that, is the one that we have. There's a little LED light inside. We're going to switch to that, and we're going to do a little bit of exploring with the Dino Light. I really need to know if which of you actually have a Dino Light. That's going to be really relevant on how we do maybe some of the next trainings. Also, when you do your quiz with your comments at the end, if any of you want a specific Dino Light training, let me know. So write that in the comments, and we can, okay, everybody's having one. Who uses theirs? How about that for a question? Got it. Okay, just because you have it, do you like it? Oh, mine died before I started. Okay. I can't say this on a recording, but maybe Gary Palmer is willing to get you another Dino Light. So don't tell him I said that. Okay, don't use it, don't like it. Okay, yeah, Adam Leonberger is a real plant pathologist, so he's got the good scopes. If you have a microscope, you're probably not going to use a Dino Light. One cool thing about Dino Lights is you USB, it shows up on your computer, and then you can share. Okay, yeah, microscope. If you have a microscope, you're probably not going to use your Dino Light. All right, but you can share, because this is USB, you can share your screen with me and we can have discussions, and you can't do that with a microscope. Oh, Master Gardener's using them. That's interesting. I'm reading the comments here. Okay, use it not for everything. Yeah, they take a little bit of practice. My recommendation is this stand, and we'll send some links. But let me share my screen. I want to go to the Dino Light site. So let's see where I'm going. Nope. Sorry, guys. Let me do this. If you can bear with me just a second. Dino Light's website there. Okay. Now I'm going to try to. Oh, geez. Do you see that, Kim? Kim is helping me here. No, I don't want desktop 2. I want desktop 1. That's what I should be sharing. Okay, can you see? There we go. There's the Dino Light website. So let's talk a little bit about what you're actually going to do. If you've got a Dino Light, you haven't used it, or if you haven't used it lately. You're going to dinolight.com, DINO-LITE. This is your Dino Light site. Going to support, right in the center tab, and then downloads. We hit downloads. There we go. Windows or Mac. I'm going to Windows. I'm assuming most of us are on Windows. There is your software and driver downloads. You need to do both, okay? You need to download the software and you need to download the driver. We have DynoLite Pro if this came from Dr. Palmer's office. And if you don't know which one you have, you can just read right on the side of the DynoLite itself. Download both of them, follow the prompts, okay? Once you do, I'm gonna get you down to my desktop and you're going to see the icon right here to the right, DynoLite 2.0. Can you see my cursor? Okay, DynoLite 2.0, that is going to be your link, okay? So I'm gonna double click that. Let me try again. Is it opening on the other screen? I've got two screens, so I'm sharing just one. Oh, it's already open. Sorry, guys. All right. So you are now seeing my DynoLite software. This is what it looks like. Kim, I need your handy dandy help and to plug that into that USB. I've got some USB ports that are not working. Okay, so your DynoLite now is going to, so I'm just waiting, it comes on, there it is live. Okay, you can expand your screen. Let's see, expand that screen by this little cursor here. Can you see my cursor there? Okay, you can expand my screen and this gets you your DynoLite. Okay, there's my hand. So with your DynoLite on the side, there is a focus knob. All right, so I'm holding the DynoLite in my hand. It is hard to steady, of course. I'm going to have a sample leaf, and there we go. Okay, so we can turn the focus knob and get what we want. Okay, this is horse chestnut. This is a Gignordia leaf blight. Or leaf blotch. I can't remember what the common name is. As you can see, we're coming in and out. So if you were having a conversation with me, we could definitely do this live. You could video it. Let's see if I can get to the top here. You see video right here. Okay, video is going to let you video record something, or photo, which is the tab that I am on. You're going to go here and you can either click this photo button here if you want, or you can touch the button on the side of the actual DynoLite while you're holding it. All right, if you want to take a picture, it's very hard to steady at this point. As you can see, I'm shaking. That's where our stand comes in. The DynoLite goes right into the stand. I'm going to lower it. And then, of course, we've got it steadied. All right, and then I can use the focus knob. I'm using the good stand, not the cheaper stand. And I can focus here and I can continue, and then I can photograph. I'm going to do that with my mouse right here, taking those pictures, and there they appear on the side. That's something that you can send to me by email, or again, we can live share screen if you want to do that. Okay? Any questions with the DynoLite? Has anybody ever purchased a better stand?
Nicole, it might be helpful to go back and have it where they can see what you're doing as far as where the focus button is, how to put it in the stand. Okay,
I'm going to unshare my screen. I'm going to unshare my screen and do that again. All right, so my stand, my good stand, so I had the DynoLite first in my hand, and I was using, and I was using it this way. This is when I was shaking. Again, this is good if we're sharing screens, okay? If you're going to use the stand, the DynoLite just presses right in. This is the focus button. It's just a turn. It just moves and turns. And then I can use this screw here and loosen, and then the shaft moves up and down. And then I tighten it, and there I have hands-free. I can use my I can use my mouse to photograph if I want to do that, or I can use this button. You see this button here? This button it's taking pictures right now. It's very sensitive. This is taking photos. Okay, so there's an LED light under here. When you plug the dyno light in, the light comes on. You don't have to do anything else. You just plug and play. Very easy once you've downloaded the driver and the software. Questions there? I'm looking at the comments here. Okay, so Dyno Light is a good shareable USB scope. It's still taking pictures if you can hear it clicking. And again, this jiggly stand is the one that you probably all have. This is the one that came with it. And this is the one that I just don't recommend. If you do want to use it, I'll get my leaf again. So you can definitely do some squeezing. This is really easy to maneuver, and you can get it down low. Okay, so you can use it. It takes some practice. It's not quite as handy as the other one, but to be able to use it, to practice using it, I'm going to challenge you that for this week, is to download the driver and the software and actually start playing with these. And when you do have something that you want to take a closer look at, to use your Dyno Light, to just familiarize yourself with it, find uses for it, because Dr. Palmer does ask very often, and he does encourage me to work with you through these. They really do have their place. I promise you that. Oh, who has the nice stand? Camille, yay, good. Glad you have the nice stand. Something else I want to point out with the Dyno Light, so you have two planes of focus. Let me see if I can share again. I lost my cursor. There we go. Let me share my screen again. All right, so there's my table. So you're looking at my hand. Okay, so I can focus all the way. I'm going to turn it left. Okay, so if you slowly move, I'm going to come into focus. I'm going to come into focus. So look at the magnification there. Now I'm going to keep, well, let's see. I'm going to keep going. And you can get, see, you can get a higher plane of focus. So there are two planes of focus in these guys. And these were actually invented for human medicine. So you can actually get, there we go, look at that magnification. They were invented for human medicine, okay? So you can really get a lot of magnification in there if you need to, just holding it very close. You see I keep snapping pictures with the button. Any questions there? I'm gonna stop sharing. Any questions? Okay, so I just really, Kim, can you do me a huge favor and unplug this thing? Okay, that's fine. All right, so Dyno Light, so your kind of homework is to actually download the driver and the software, the updated software, and then to play with it. And if you want to work with me, I'm very happy to do that with you. We can, again, do it by Zoom or by Skype. Really easy. If you want to practice with each other, it doesn't matter. I highly recommend that you make that a habit. Remember last time we talked about basic tools. Your hand lens is never, ever far away. So looking at samples at your desk where there's good light above, going closer to a window. These are lighted, the ones that we give you. If you don't have one, let me know. You're also going to get them at the practicum. But when you use your hand lens, you put it up to your eye, just like your eyeglasses go to your eye, and then you lift the sample to yourself until you get there. Okay? So I'm looking at apple scab on a crab apple, but I'm able to see it this way. So hand lenses always in your pocket. I recommend that you have more than one in your desk, one in your car, one in your sample bag, always having hand lenses. My emergency are these credit card magnifiers. We gave you these as well. They're very thin. They can fit right in your wallet. They're the size of a credit card. These say plant pathology, which makes them even more cool than any of the others that you might buy. So having these, I keep this in my phone case. There is one in my purse, one in my tote bag. So this is an emergency. This is also when I forget my glasses. So we'll be giving you these as well. If you don't have one of these, again, shoot us a message, an email, not through this chat because we'll lose it. It'll scroll out. Okay, one more thing before we get off of a dyno light. I want to show you something really cool. This is a backlight. It comes with a cord. It lights up, and this allows you to double the dyno light as a compound microscope. This backlight alone was like $450. It was expensive, but we will, when we work on bacteria and some other things, we're going to use this for upcoming webinars. But this is kind of a really kind of a on the tailgate kind of microscope, kind of really portable, but this backlight really will let you double as a compound scope. So it's a good training tool. It's good for your master gardeners. It's good if you're kind of in a pinch. Very few agents have both a dissecting scope and a compound scope. This is really nice. Being the new budget is coming up, and then you would still use your same dyno light, and then you would use the backlight, and then you would lower, okay? And then there's your compound scope with the screen being your viewfinder, okay? Questions there? Okay, and now what time is it? Okay, so now one of my favorite tools is what we call a proscope. Your proscope, again, an LED microscope. It's portable. It's got the USB plug to charge it up. It doesn't require batteries. It just charges. Mine stays plugged into my computer, and when I need it, I just unplug it and take it away. The proscope, we're going to give you that link to get to the proscope. It comes with a phone case. So, okay, backtrack. These are going to align with the camera on your phone or on your iPad. All right, so they hook. You can see there, let's see if I can turn it. There are two hooks here, and it just hooks. Okay, maybe I need my glasses. It just hooks, and then your phone clips in. This one is the telephone one, cell phone, I'm sorry. And this one is iPad. I like to use the iPad. If you ever see me around training, I will use the iPad. So it's got 2 feet, and then the proscope serves as the other. And then my iPad, so my iPad camera is going to align with the proscope, and there it clips on this way. Okay, and then turning on. Okay, so you turn on your proscope on the side. I know I charged it this morning. There we go. You see the little LED lights came on in there. Then I turn on my iPad. Okay. And I'm logging into my iPad. And then I'm using the camera. And then there it is. The proscope is using your phone camera or your iPad camera. Okay, I'm going to try to hold this up and get this to work. So it is self-focusing, just like the camera. There we go. And there we go. And then you can zoom. I'm going to try to do this for you. You can just use the touchpad. You can touch to focus, just like you would. There we go. So you can use that to focus if you want to see anything. Let's see if I don't make a mess here. Okay, and then you could take the photograph and you could share that. You could share those right in the field if you're using it. Okay, no, they will not fit over a case, Philip. You've got to take your case off, which is why I use the iPad, because my phone is in a case that's really hard to get out. But this is something that is really easy to use. I really like them, and they're good. The iPad version is really good for training, depending on who you train. But the phone version, and even in the field, when you have a stakeholder, they want to see. I keep moving here. But you're really steadying here on the desk this way. You're putting your proscope right on the leaf, and then you are zooming. okay? And then photographing, videoing if you choose, and then you can share it right with us if you need help, like immediately. Okay, who has a pro scope? We're gonna ask you that when we give you the survey at the end, just kind of have an idea. Oh, okay. iPhone 6 is where they stop. I know at one point they were not doing, I have an Android, they were not doing that latest Android. So it just kind of depends. They're a little bit slow. Now, if you want both, so when you buy the Pro Scope, you get one clip. So I bought mine with the iPad clip, but I bought the phone clip separate and alone for like $30 or $35. So you're looking at $150 for the Pro Scope plus one clip, and then the additional clip was I think $35. So I used to toggle between the two. My phone, I have a different phone now. So this one is for my old phone. I don't use it anymore, but this is a really good tool as well. So my two favorite tools, I guess this would have to be almost tops, and it's a good way if you use it with the iPad. It's a really good way to show your stakeholder exactly what you are seeing. And sometimes, you know, with your stakeholders who don't quite trust, or they insist on having something sent off, then you can show them, look, these are the structures, this is what I'm looking for, this is why I think it's a pathogen, this is why I don't think it's a pathogen. And that kind of gives you a little bit of a validity there, that if you have a skeptical stakeholder. All right, questions here. We're going to send you these links. So $150 for the set with the Pro Scope. Dino lights you should already have. If you want a better stand, then those are about $100. We're going to get that to you. And then the backlight to make a compound scope out of the Dino light is about $450. That's expensive, and that's not something you really need. Just if you're kind of like plant nerd and you really want to do that. Questions? Kind of finishing a little bit earlier than I bargained for. Okay, so advanced tools. So once you're, let's backtrack a little bit. Once you're in your office, either a walk-in or a field visit that you have done, you get to review the information. You get to go through that checklist. You're eliminating the obvious. Okay, you're eliminating anything that might be string trimmer damage, or the kid with the hatchet, or herbicide drift. You remember last week we talked about looking for patterns, figuring out what's going on. Once you've eliminated that, and even if it's a walk-in, you still need that back information. So once you have decided that this may be pathogenic, then you've got to decide what is the right sample. We talked about that again last time. The right sample is going to give you, and of course, leaf diseases are minimal of what we deal with. So we like to show you leaf diseases. They make really good examples, but root diseases or root problems, compaction, abiotic type things, anything on the trunk or stem, all of those things, usually they come in multiples. We rarely get just one thing that's a causal agent. It's usually a combination of things. So to be able to eliminate all of those things, have an idea of what's going on, to be able to look at multiple plants, at multiple plant parts, not just one single sample, gives you an idea. Like with this horse chestnut, we know this is Gignardia. So getting it to the diagnostic lab is not going to tell you much more than what you can see for yourself. Apple scab, this was one that, I mean, we could talk about those in the fungal session. So again, leaf issues, we'll be looking at stem and root problems as well in the near future. And to be able to at least take some pictures. Sometimes you as agents don't have to get to the primary diagnosis, but you are going to eliminate the obvious. Sometimes a grower needs to know if there's something invasive or if there's something really critical and they need to manage it now. you can get to that point, and then that'll give your sample time to route through the diagnostic process. This is a tough time of year for diagnosticians. Again, about 3,500 samples a season, and they all really, the majority of them come in like a 3-month period. Yes, you can absolutely use the ProScope. I'm reading a comment. You can use the ProScope to send images to us by email or by text or whatever, because it's using the camera of your device. Okay, so you can attach it to an email, you can text it. I know Lee Townsend used to accept a lot of text with insects. He's not there anymore. If I remember right, Rick Besson is having problems receiving images through text messages, but you can put them in emails. Don't forget to attach them. Don't let the phone embed them. And that's really helpful. If you even FaceTime, you can't FaceTime me because I have an Android phone, but if you make kind of an appointment with a specialist and you want to FaceTime or something from your phone from the field, you can absolutely use that, that ProScope to do that right there live. You can use your, if you bring a laptop with you, you can use your Dino Light in the field as well, but it would require a laptop. Because of that USB connection, you can't really use it on your phone. Okay, so you have the option of using some of these tools to kind of drill down and get to the bottom of a lot of issues that come into your office. We have some tools, and then there are just some really simple ones like hand lens, magnifying glass, whatever you need, just to be able to process all of the information. And again, sometimes that's a little bit harder to do in the field if you're not alone. Sometimes you have to go back to the site. If it's a commercial sample, if it's anything you question, we recommend a sample go to the diagnostic lab, but you can still work on your own set of samples in the meantime. That's totally okay. In fact, we encourage that. And that's the way you learn. If you really want to get some practice with this, taking a lot of notes, you're not going to see as much as we do because we are focused, so we see it constantly. And whereas agents just don't see it at the same rate. So take lots of notes, take pictures if you want to do that. Our fact sheets, remember our website, looking at our fact sheets, Kentucky Pest News newsletter, receiving KPN will tell you what we're seeing, what to be on the lookout for, and that helps you eliminate some of that obvious, because we tell you what's going on and what you should be seeing. A lot of social media sites. I have a social media, a Facebook, Twitter, and Google Plus that is out there, and we do send alerts through there. Emily Pfeiffer does vegetable diseases, so both of us have articles, or Kim Leonberger rather, has articles in Kentucky Pest News. Again, what to look for, what's going on. And you can always refer back to fact sheets on the Department of Plant Pathology's website, Department of Entomology's website. And that'll kind of get you a little bit closer. Okay, any other questions? All right, so let me share screen again. So remember that in order to get credit for this program, you've got to complete the quiz. The quiz was really easy, if you remember right. Oh wait, sorry, I'm sharing my Dino Light. That's not what I wanted to do. Here we go. I thought I was. Uh-oh, wait, hold on, guys. All right, I'm sharing the wrong screen. It is, I might need to get up. There we go. So our training website, this is where we're going to have the resource guides. And this is the quiz. We're going to send this link. Kim, are you posting that link? Okay, Kim is going to send the link in the chat box. This is the link that you are going to use to take the quiz. It's just a 5-question quiz. This is where you're going to put your name, and that from there, it's that list of names at the end of the quiz that we submit to Kevin Horn for credit. Don't forget to go that far. There's a comment box for you if you want to give any comments. what's coming up next, what you didn't understand, etc. Oh, by the way, this is an underscore here. So I guess I need to remove the hyperlink so you can see it a little bit better. Okay, that is an underscore. And you can, again, let us know if you can't access it. This video will be available for 1 week. Any agent who could not participate today is able to access it for a week. We have 7 total webinars plus a practicum as part of the certificate program. Anybody can participate in any individual one to get CEUs. That's perfectly acceptable. Any questions? I hear someone. There's a question.
Could you run through your 5-picture rule again?
5-picture rule. Okay. 5-picture rule is the entire landscape, and I say landscape or field, so surrounding plants as well. So the wide angle view, the entire plant in full view, soil level, so the crown of the plant, soil and surrounding weeds, whatever. So ground level. Number 4 is looking for any cankers or any stem lesions or anything that might be a common denominator within a problem, like say a branch. And then finally, leaf close-up. That's the 5-picture rule, and we will post that again. Aha, wow. Oh, that's Kim doing that. Of course Kim's doing that. Okay, we'll get that back out to you. And sometimes you may have more than one canker, sometimes you may have more than one leaf picture. But you're never gonna overload an email box. You're never gonna zip a file. And that kind of forces you to take a better picture. Double check your pictures. Don't send blurry pictures, please. That doesn't get us anywhere. That just is delaying your grower to get the answers that they need when we have to reply, and then you have to do it again. And the longer we take, the longer your stakeholder is waiting and worrying. Okay, any questions? All right, we're going to be sending the resource list out to you with all of these links that you need. Yes, Linda, we're going to cover fungi next, and we're going to look at fungal structures. And I'm actually going to use these tools, and I'm going to share my screen with you. So you're going to see that. So we're going to play with moist chambers. We're going to look at in and out of moist chambers. We're going to look at some of the big structures. We're going to look at situations when we don't see a structure and what you can do with that. We're going to look at roots. We're going to look at stems like cankers, and we're going to look at maybe some wilts, how to dissect. Next Friday, we start fungi. Less lecture. I'm going to be sending you reading materials, less lecture and more demo. So there's going to be kind of some pre-work with the next ones, and that'll kind of help you understand. Those materials will probably just go out the day before, simply because I want to be able to review and collect samples and get the right things to you. So look for those. The earlier you can sign up in KERS, the better. That way I'm using that link. I'm using the KERS email list in order to send these resources to you. So fungi are next. We're also going to cover the oomycetes, which are the water molds. They're separate. Bacteria. I can't remember what else.
Viruses and nematodes.
Viruses and nematodes are going together. We don't see quite as many of those. And then our last one are going to be abiotic issues, things that might look like disease but are not, that are easily confused. Because remember, about 66% of the samples that I touch are really not disease at all. So abiotics are really important. Okay, any other questions? You're always welcome to call me or email me. Texting is a little bit delayed for me because if I have a keyboard, I'm probably not looking at my phone. 66% is an unlucky number. I'll have to think about that. At least there's not another 6 in there, right, Andy? Okay, 67%. How about that? That sounds better? Maybe 70. So we will be a little bit more specific in the future. You can hit and miss. If anybody wants a video, in hindsight, it can't be part of the certificate program if it's too late, but we're always going to want to share these with anybody who needs them. We're going to get a little bit more specific next time. The first two webinars were more playing, but just kind of review. So we're going to keep it light, but we still want to be able to get you the information that you need. So email me, call me anytime. Call my cell, not my office, remember, because I'm rarely here. You're anytime welcome to call or message me. If you're heading out to a field visit, you can message me in advance and say, what are you doing tomorrow at 2 o'clock? I would like to be able to connect to you while standing in the field on a field visit. You're always welcome to do that. And if you've got a grower over your shoulder, you can kind of give me a little bit of warning, and we can kind of make that talk appropriate as such. So don't hesitate, and we will see everybody next week for fungi, the biggest group of our pathogens. So thanks, everyone. Again, this is recorded, and we will chat later. Thanks a bunch.